As you are watching this video, the cells in your body are dividing and the DNA within them is being meticulously copied. Every day, millions of mutations occur inside your body during cell replication. Contrary to popular belief, cell mutations are not always harmful. According to the National Library of Medicine, a gene mutation is a permanent alteration in the DNA sequence that makes up a gene. A point mutation is when a single base is exchanged for another base. A transition point mutation is the exchange of a purine for a purine or a pyrimidine for a pyrimidine. A transversion point mutation is the exchange of a purine for a pyrimidine or vice versa. As a reminder, the purines are adenine and guanine, and the pyrimidines are cytosine and thymine, and uracil instead of thymine in RNA. There are different types of point mutations that have different effects on the amino acid sequence. Given the following sequence, the first step is to change all of the thymine bases into uracil bases because it is the RNA that will be translated. This is the original strand of RNA and the translated amino acids. A Missence point mutation is when the exchange of bases causes a codon that codes for a different amino acid. This can alter protein function, making it more or less effective or result in a non-functional protein. This can also be neutral and have no result on the protein function. Using the original strand, it can be seen that the guanine was substituted with a uracil, a transversion mutation because it was a purine exchanged for a pyrimidine. The amino acid cysteine changed to phenylalanine. This mutation can even lead to amyloid diseases in some cases. A nonsense mutation is when the exchange of bases changes a codon into a stop codon. In this case, the second trinucleotide was altered into a stop codon. The guanine was substituted for an adenine, a transition mutation because both were purines. This type of mutation causes translation to end early and the protein to be shortened. This frequently results in a non-functional protein. In this case, the tryptophan was changed to a stop codon, creating a non-functional protein. A silent mutation is when the change in nucleotides still codes for the same amino acid. Looking at this strand, the uracil was substituted with a cytosine, a transition because both are pyrimidines. This mutation has no effect on the protein function. The amino acids remain the same, as does the protein. The final type of mutation is a readthrough. This is when the stop codon is changed into a codon that codes for an amino acid, resulting in a longer protein. In this case, the stop codon was changed into the amino acid glycine. This would create a longer protein that could alter the function of the protein. This is an example of a transversion because a pyrimidine was substituted with a purine. Mutations can be harmful, harmless, and even sometimes beneficial. It is important to understand the different types of mutations and their effects.